like to talk about pharmacogenetic kit that we are developing. And in continuation of the Subu presentation, there are some facts on pharmacogenetics. For example, in US, there are more than 100,000 deaths and 2.2 million serious side effects caused by adverse drug reactions every year. And in recent years, this situation, these numbers are decreasing because of introduction of pharmacogenetics. So it is well known that information on the patient's pharmacogenetic status minimizes the occurrence of the side effects. And the most important time points was in 2007 and 2010, when the pharmacogenetic community, when the FDA has approved the usefulness of the genetic information for the such drugs as Warfarin and Clopidogrel. And that was really important, not only for US pharmacogenetic community, but for the whole world. And we are, in our research, we concentrate on the Central Asian region. You can see it on the map. So it is pretty big, and the Central Asia, it's about 66 million of people. There are countries that ends with a stan, for example, Kazakhstan, Uzbekistan, Kyrgyzstan, Tajikistan, and some others, but excluding Afghanistan and Pakistan. And it's curious that the stan, it translates from the Persian language as a country of. So, for example, Kazakhstan, it means the country of Kazakhs, and the Kazakhs are the population that lives there. So what we noticed when we started our research, that pharmacogenetic testing was not conducted in the Central Asian at all. So it was completely absent. And our small company that was established in 2012 decided to focus on the pharmacogenetics. And we have a really big problem to solve. So the problem is complete unawareness about pharmacogenetic testing in the region, and absence of the testing. And our goal is to introduce the pharmacogenetic testing and to the health centers and hospital. First, we want to start. We already started with the Kazakhstan, and then we want to expand to other countries. And we received some funding from the World Bank because the problem is very topical, and also from the ministry, from the local Ministry of Science, to develop such a kit. Then we already started last year to provide some services to the national centers in the capital of our country. So we decided to focus on cardiovascular drugs, because cardiovascular disease is one of the leading causes of morbidity and mortality in Central Asian region and in Kazakhstan specifically. The reason is that introduction of this testing will minimize the side effects and complications. And on this chart, you can see that morbidity is increasing every year by 50,000 new cases. So what are the benefits of pharmacogenetic testing that our country will experience? It is the increase of lifespan and decrease in mortality when the cardiovascular drugs are used when corrected individually. Then economic impact, for example, warfarin is prescribed for a lifetime, and decrease in its treatment is very significant when it's corrected individually. And also the whole healthcare will benefit from this. As you know, as already Subu mentioned in his presentation, there are ethnic differences in pharmacogenetics, in pharmacogenomics. And sensitivity of various ethnic groups to the drugs is different. And when we started this research, we tried to find some information on genetic, on the frequency of alleles in biotransformation enzymes in Central Asian population, but we couldn't find a single work or review on this topic. So our next step was to study this by ourselves. And we figured out that Central Asia is the one is among the least studied regions in the world in terms of frequency of different alleles in biotransformation enzymes. And historically, Central Asia has always been a crossroad between West and East. For example, maybe you remember the Silk Road and Genghis Khan invasion and some other events that happened in the history. And so the studies of the region that we conducted indicate that the population of Central Asia has a mixed genetic composition that is intermediate between Asian and European population. But how we got this information, we did the expeditions last year and this year. We went to different rural regions of our country, and the country is really big. It has a 9th area by size in the world, Kazakhstan. So we went to different expeditions to different parts of the country and collected around 1,000 samples from Kazakh population. And then we genotyped, we did the genotyping with the real-time PCR in order to figure out the frequencies of different alleles in biotransformation enzymes. So this is just an example of genotyping on one of the CYP2C19 variant. And we got this table, and we still continue this work, the genotyping. It's a lot of work. So as you can see, the number of samples that are genotyped is different because we couldn't finish it to this time point. But this is preliminary data that we compared to other populations. And as we can see, so we did the comparison to available databases. For example, it's data from 1,000 genomes for Asian population, then Caucasians, and so on. And we figured out that frequencies are of some alleles are intermediate. For example, if you look at CYP2C9, it's 0.2 for Kazakhs, then almost 0 for Asians, and 0.12 for Caucasians, so it's somewhat intermediate. Or for example, look at the of course C, vitamin K epoxide reductase, it's 0.7, and for Caucasian 0.4, for Asian 0.9. So this shows like intermediate position. And based on this data and from the data available from other kits and panels for genotyping, we did the following conclusions: that for correction of for dosage of warfarin, we need to genotype these alleles of these enzymes. It's CYP2C9, of course C epoxide reductase, and CYP4F2, and gamma glutamyl carboxylase enzyme. And also for Plavix, the following alleles are needed to be genotyped for individual dosage of Plavix. And this is how the kit looks like. So it's based on real-time PCR. Also, we do the real-time PCR and then see and then check the dosage online with well-known software, internet-based software, warfarindosing.org. And we already started the clinical trial, so we started to correct dosage in 2 clinics that are based in the capital of our city. So 136 dosages were already corrected. And this is it. Thank you for your attention. And yeah. And just the main message of my presentation is awareness, pharmacogenetic awareness. Because, for example, in USA, I think it seems normal to do pharmacogenetic testing, but in other countries, people, clinicians, and healthcare institutions, they even don't know that pharmacogenetic testing exists. So it's very important to bring this information, to introduce pharmacogenetic testing to clinics in developing countries. Yeah, thank you.
I have a quick question.
Yeah.
Is this a multiplex PCR?
Yeah, it's multiplex, yeah.
Not easy to validate.
Well, yeah. Okay.
Hello? Pavel? You're
Yes.
on. Congratulations. Yes, I'm a population geneticist, and as you point out, there's been a long-standing deficit of human population genetic data. So congratulations, you made a great start. Mm-hmm. And I wondered, of the samples that you've collected, are they broadly consented, where you could collaborate with people that might have funding to do additional genetic tests, such as a whole genome SNP chip, or?
No, we don't collaborate with anybody. We did it ourselves.
But do you still have the samples, and
yeah.
Are they broadly consented? Are they consented?
What do you mean by broadly consented?
So, when the samples are taken,
yeah.
you have to have consent.
Yeah, of course.
Okay.
Every person signs the
right.
consent, and it is approved by the ethics committee.
Right.
Yeah.
So do you think the consent is broad enough to allow additional genotyping for like a whole genome biobank chip? Or
maybe.
If they are,
yeah,
I will put you in touch with some people who would probably be interested in collaborating.
I think so, yeah.
I have a quick question. Is there a lot of consanguinity in the population? Are they marry among relatives, marriages? Is there a lot of inbreeding in the population?
Inbreeding? Well, no, not a lot, because it's a nomad population, and it is so people used to travel a lot, so there is not a lot of inbreeding.
No, there are no isolated groups in the mountains?
Very, very small, because isolated population usually we can meet on the islands or somewhere else, or in the mountains.
Yeah.
But it's a plain, and it's a pastoralist population. So
I thought you have a mountainous area. Isn't there a lot of mountains there?
Mountains, no, not a lot. Only on the border with China,
yeah,
there are mountains, but it's a flat, it's a steppe, and a lot of movement.
Right.
There.
Right. Okay.
Yeah.
That's fine. That's all.
Any more questions? Thank you.
Mm-hmm.